Okay, this is a voice recording for the nervous system, specifically the cellular component of the nervous system. So there's another video that goes with the macro, talking about the brain and the different senses, but this one's just going to be about the main worker of the nervous system, the neuron, and its supportive cells. So let's talk through what neuron parts are, or the anatomy of a neuron. So general star-shaped here. This is going to be one that's a multipolar. There's bipolars as well, and unipolars. So here we have, at the star end here is the cell body, and I'm drawing out a little bit of the axon here. I'm just going to draw one synaptic bulb, even though there are multiples that come off this way. So if you wanted to, you can extend out like this. But I'm going to focus on just one enlarged synaptic bulb here. And then on this end up here, we have all the dendrites that reach out and touch different things, whether it be a muscle or a gland, or if they touch different neurons. But here you have all the extended, so this is all dendrites. And here we have the cell body, right, where we would have the nucleus. Sometimes it's called the soma, the cell body. Right here, a very important spot to take note of, this area right here where we end the cell body and we start the axon is called the axon hillock. And we'll talk about that as far as graded potential versus an action potential at the axon hillock. Most of the cell's workings as far as cellular respiration, things like that, mitosis, all the DNA synthesis and all those pieces all happen right here in the cell body. Dendrites, like I said, are going to be the things reaching out and touching different cells. You could also have different synaptic bulbs coming in and touching the cell body or different areas of the dendrites for signal being retrieved. So the signal is always going to move from the dendrite area out to the end of the axon down here at the synaptic bulbs. The other things that we have to keep note of is these little fat sheets that are right here. These are myelin sheets. And myelin can be formed by two different types of cells. If it's the central nervous system, it's going to be oligodendrocytes. And if it's the peripheral nervous system, it's going to be Schwann cells. Now, you can see the gaps. I've over-exaggerated these gaps. These gaps are very, very small. But there are gaps here in between the myelin sheets. Those are called the nodes of Ranvier. And those are going to be important when we establish what's called continuous conduction of signal and what's called salutary conduction of signal. Now, these areas are going to be the areas that depolarize. They won't depolarize at the myelin sheets because of that barrier to the surrounding area and the inability to have gates. We'll talk about those gates. So it's all going to be electrical moving down this neuron, and then when it gets out to this end here, we'll talk about generally we end up seeing some sort of chemical being secreted, and we'll talk about those to tell the next cell what to do. So let's continue our journey, moving on to the next. Okay, so let's talk about how this cell is able to do what's called an action potential, or a cascade or a jumping of signal down the cell body all the way out to the ends of the axon. So let's establish what's going on. If this is the inside of the cell, and this is the outside out here. Outside here, we'll have a lot of sodium ions on the outside, and we're going to have lots of K+ or potassium ions on the inside. Now, even though those are both positive, generally the inside is much more negative due to other anions that are in here. And it's going to be more about the changing of the sodium coming in and the potassium going out that's going to guide the actual charge inside the cell. So when we take a reading, we have little mini volt readers. We could put a mini volt reader inside and outside. We can establish, and based on the inside compared to the outside, we say that the inside's negative. That negative is around negative 70 millivolts. So that's your resting state. So at resting state, both your sodium gates, there's little proteins here inside the cell membrane that are going to block transmission, because you remember this is phospholipid bilayer here. They're not going to let things just pass through, especially things that are charged. So gates have to be in there in order for these things to move in and out. So there's going to be gates that allow for sodium to move specifically, and there's going to be gates that allow potassium to move specifically. And then there's going to be some other ones in here that are pumps that help us to reestablish. So they're going to be moving sodium, potassium. Those are called sodium potassium pumps, and those take energy in order to run. So let's talk the resting state. So right here at 1 is the resting state. And we have negative 50 on here, and specifically negative 55 is generally the one we talk about, is what's called the threshold. So we can have signals coming in, and we can have positive readings, sodium may be rushing in, but if we don't reach that negative 55, it does not do this whole spike. So every single neuron is going to fire at the exact same shape, regardless of stimulus coming in. So even if you have something that's a strong stimulus or a weak stimulus, this curve does not change. It's all about the frequency of the curve, and then how many other neurons are being allocated to respond to whatever it is that we're looking at. So here's the breakdown. Oh, too far, sorry. Here's the breakdown of the different gates. So here, when we're at resting state, you can see the sodium can't get in, the potassium can't get out. We have that negative inside, positive outside. Then we start depolarizing. That means we hit the negative 55 threshold. We're going to depolarize. So notice what happens is the sodium rushes into the cell, and that's because there's more sodium outside than inside. So based on entropy, things are going to go downhill, right? They're going to go down the gradient. And so sodium rushes into the cell, making the inside of the cell more positive, hence why the graph starts going upward. Once we start getting to the repolarizing, that means that the sodium gate closes and the potassium gate's open. So that's right at the top of the peak up here. And then we're going to start moving downward because potassium's rushing out. So positives are rushing out. And that's because there's more potassium inside the cell to start than outside. So it rushes down its gradient, and we make the thing more negative again. And we have this undershoot where the potassium gate stays open longer than it technically kind of should. And the reason for that is it allows for the neuron to make sure that it doesn't cause itself to refire. So it's a kind of a safety net, so to speak, so that we don't end up getting continually firing from the same impulse if we don't want it to. So it's kind of the reset. So consider undershoot as the reset, making sure that we don't go too far beyond where we need to go. All right, so let's talk about this whole action potential piece. So if we get that negative 55, here's a breakdown of how this is happening. So if we're talking about continuous conduction, so continuous conduction is literally just moving straight down the axon all the way down to the synaptic bulbs, no jumping whatsoever. We're going to have sodium rushing in here, and you can see the sodium rushing in is going to cause the next section over here to depolarize then. So then this section starts depolarizing and gets to the negative 55, and it causes the sodium gates to rush in here. And as this one's starting to kick, this one is in the undershoot. So even though sodium is rushing in and pushing backwards, because it's in that undershoot, it won't cause this back area here to refire again. So that's one of the big purposes of the undershoot. This one then causes the next section to depolarize, the next one, we keep going down. Now, if it's solitary conduction, which I'll talk about in a second, instead of going literally touching regions, we can jump regions. It can move a lot faster as it moves down that axon. So here's where the undershoot's important. So if I actually stuck something into the axon here to depolarize it artificially, there'd be no undershoot established because everything starts here at the axon hillock. There'd be no undershoot established. So actually, we would get a signal going this way and this way, as opposed to it starts here and it goes downward. That ensures that the undershoot is right behind where it took off from. Reminder that different signals coming in, we'll talk about this a little bit more, can cause what's called a graded potential. So we have some things that would be EPSPs, or excitatory postsynaptic potentials, meaning that some cells will cause this to excite, and then we have IPSPs, which are considered inhibitory postsynaptic potentials. So they'll cause this cell to get more negative, and it cause maybe a washout of excitatory. So that's your graded section. So we're grading, we're summating whether or not these two adding together is going to get us to negative 55 by the time we get to the axon hillock. If it is negative 55 or above, then we actually cause this whole fire. So continuing on, here is the talking of the one neuron to the next neuron. So this is an example of chemicals or synaptic connections. So we have the nerve impulse all coming down electrically, the way that we were just talking about, as it gets down to the synaptic bulb. So here's the end of the axon. What happens is, as this electric signal or the sodium rushing in, it's actually going to cause little portals at the end of the axon to allow calcium to come in. What calcium does is, there's neurotransmitters made down here. So we have maybe some ER, some ribosomes, whatever it is that we're making, depending on the neurotransmitter that we're focusing on, makes that protein or makes that steroid type of element. And those things, when the calcium comes in, causes it to form a synaptic vessel. And when it touches up with the plasma membrane here, we get exocytosis. We get the neurotransmitter pushed into the synaptic cleft. When it pushes into the synaptic cleft, then it binds with the effector cell, the next cell, and there's these little ligand receptors. And these ligand receptors are very specific to the neurotransmitter that's being made. When it binds up, it can cause this cell to open sodium gates. And when sodium rushes in, it could cause the next cell to have an action potential. They might also cause this next cell to open a potassium gate, which means that it's an inhibitory signal. So depending on what type of gate it opens, is big. The last piece is, once this thing binds in, the only way to stop this signal is to destroy the thing that's affecting it. So the neurotransmitter has to be degraded. So what this is showing is the neurotransmitter being degraded. When it gets degraded, the gate closes again. So there's different types of things. We talked about with the muscle unit, a specific neurotransmitter called acetylcholine. So acetylcholine release is going to cause the muscle to fire. So if this cell right here is a muscle cell, it's going to cause gates to open and allow for calcium rush in and allow the actin, myosin to do their thing like we had talked about with troponin and tropomyosin. But this ACH then needs to be degraded. Otherwise, the muscles continue to fire and you get something like tetanus. So there has to be an enzyme, and the enzyme is called acetylcholinesterase. And acetylcholinesterase will destroy the neurotransmitter, closing it and allowing for this muscle to get re-stimulated if another signal gets sent, as opposed to continual stimulation just because it's bound up here. Another way that these neurotransmitters can be removed from the site is they can freely diffuse out. We also have neuroglia that can help absorb or break down these neurotransmitters, or the neurotransmitters can be reuptaken by the presynaptic neuron. So we have the presynaptic neuron, we have the postsynaptic neuron. This one's going to affect this one, and then we have, like I said, the different types of molecules or events that can cause those neurotransmitters to get away. So when we looked into, and I say we loosely, like I'm not the one that discovered this, but scientists, to figure out why people have depression, they linked it up with dopamine, the neurotransmitter, and they found that the dopamine wasn't either A, being produced enough, or it wasn't staying out here long enough. So a lot of the drugs that are out there, Prozac, will keep the neurotransmitter in that synaptic cleft longer to allow for more stimulation and allow the person to feel better. All right, so let's talk about the EPSPs and the IPSPs. So here we have an input from maybe another neuron causing a sodium gate to open, but you notice that the firing isn't frequent enough, so it opens it and then it opens it, but it's not enough to get over that threshold, so there's no summation or there's no action potential that fires at that axon hillock. Whereas if the two signals come in quickly, then if they come in quickly, it gets it over the action potential or over the threshold, and the thing fires. We could also have two signals coming in, adding or doing a graded piece, and when they grade together, the summation allows for action potential to be reached, or the threshold to be reached, and the action potential to fire. The opposite, or another scenario I should say, is when an excitatory comes in, but then an inhibitory comes in and we end up washing it out. So the inhibitory brings it more negative, so it might be opening a potassium gate, and the excitatory is opening a sodium gate, and the two wash them out. We never get an actual summation that causes an action potential fire right here at this axon. So that's EPSPs and IPSPs. And the last couple pieces that we need to connect in is how all these signals are firing. So one of the ways that your brain is able to work with stuff is obviously senses coming in. We'll talk about this with the next section. And then how that coordinates up to your brain. But sometimes you have reflexes. And reflexes is when you have a signal come in, it hits the spinal cord and it comes right back out. So example, when you're sitting at the doctor's office, you're sitting on the chair and here's your leg hanging off the chair, right? And they have that little hammer thing. That's a horrible leg. They have that little hammer thing that has, right? And they hit your knee right here. They're hitting the patellar tendon here. When they hit that patellar tendon, it causes your knee to jerk out, right? They're inciting the patellar reflex. So what's happening there is you're getting the sense in, it's going straight to your spinal cord, and it's coming right back out to your skeletal muscles, causing you to feel. That's a normal response. It's important. So they're just checking to make sure that all your circuitry is working properly. If it's not, then obviously there's some things that they need to look into. Last couple pieces as far as the nervous system goes is looking at inputs versus outputs. So sensory is going to be your input piece. So we have sensing the internal environment and the external environment. Both of those come in. That's part of your peripheral nervous system, right, away from your central. Remember, the central nervous system is your brain and spinal cord. Peripheral nervous system is pretty much everything else. So in the peripheral nervous system, we have the sensories, or the things coming in, and we have the effects, things that we want to do because of the senses that are coming in. So we have the autonomic, or things that we don't have to think about. They automatically are done. And then we have the somatic, the ones that you control or think about doing. So in the autonomics, we have two guided systems. One is called the parasympathetic, and one is the sympathetic. Consider the parasympathetic the feed and breed, right? So feeding and breeding. Basically, you're chill and relax. If you're eating or if you're breeding, this part of the system is kicked in, and you don't have to think about it. There's all kinds of responses that go with those pieces, like sending blood to the digestive system when you eat something. That's the feeding. Or the sympathetic, which is your fight or flight. And we'll talk more about this when we get into the next section. But fight or flight is exactly what it sounds like. The part of the system that's going to send blood more to your muscles so that you can respond to some sort of stressor that's coming about. And we'll kick this back when we come into the endocrine system later on this year as well. So understand the divisions of the nervous system, especially the cells that kind of participate in it. The last piece is the support cells that will be on your test. So you obviously have the neuron, the main worker, but then the neuroglial cells are the support cells. So you have the astrocytes, which are going to help form the blood-brain barrier, the ependymal cells, which help make the fluids that make up your cerebral spinal fluid, or CSF, to help bathe your neurons in the proper fluids. We don't want the blood to intermingle with that fluid. That's very bad for you. Microglia, which act like white blood cells almost for your nervous system. They're going to help clean up debris and junk that might be forming in that area. And then we have the ones like this is showing that make the myelin sheaths. You're seeing the wrapping of fat around the axon. On the central nervous system, they're called oligodendrocytes. But in the peripheral nervous system, they're called Schwann cells. They form the same exact myelin sheathing and same types of nodes of Ranvier to allow the signal impulses to travel much faster. And there you have the entire nervous system cellular, or I like to say micro section of notes. Wish you best of luck with studying.